Okay, we know about independent recombination, of genes, and now we I gonna talking about linkage of genes. There are two main types of linkage genes. The first type is complete linkage, and the second one is incomplete linkage. What is the difference? First of all, look to the complete linkage. Okay, when or if complete is linkage, there should be parental combination only and no recombination. Morgan in 1919 reported a complete linkage in Drosophila. When ordinary male white fly with gray body, write it, gray body, and normal wings, was crossed with female having black body and vestigial wings. This is black and vestigial wings in F1 generation or F1 hybrids were all gray-bodied and normal winged with dominant characters. This, scrolling down, and we have this is the male organism and this is the female organism, male and female. And in generation 1, F1, we have an organism with dominant characters, and this organism are with gray body and normal wings, okay, gray body and normal wings. Okay, but we are talking about complete linkage. But in when F1 generation, this organism with this genotype crossed with homozygous recessive female with this genotype, they are give in generation 2 to Mendel the next individuals. Okay, there are two types. We have this organism can give us only two types of sex cells with only two types of genotypes. Delete it and come back. This organism can give us first sperm with both capital AB with dominant genes, and this spermatozoa can give us only with small ab with recessive genes, so only with recessive genotype, because these genes are linkage between them, and these genes too. This organism can give us only one type of eggs, and with recessive homozygous genotype. Okay, crossing. Make a Punnett square. And here we have AB genotype, female, and this male genotype, AB capital, AB small, A, B, and A small, B small. This is 50% to 50% in generation F2 or F2 generation, because these both genes and these both genes are linked between them, and there are in this case we can see working on second Mendelian law, because these two genes go to the next generation together. They still stay together in all generations. Okay, the next one is incomplete linkage. Second. Incomplete linkage. What is the incomplete linkage? Incomplete linkage produces new combinations of the gene in the progeny due to the formation of chiasma or crossing over in between the linked genes present on homologous chromosomes. You know that when is inside the meiosis in prophase 1, we can see very interesting process, and name of this process is crossing over. It is crossing over. What does mean crossing over? Crossing over, it is when some parts of homologous chromosomes change their places. For example, this part of homologous chromosome 1 cut from the arm of chromosome and go to the arm of the second chromosome, and homologous part of the second chromosome must go to homologous part of the arm of the first chromosome. Okay, and the action of second Mendelian law, this process, the process of crossing over, It is the base of the second Mendelian laws, because this process makes a recombination of new genes, of new genotypes. I'm sorry, of new genotypes. Okay, but in our sex cells, when they are forming, crossing over can be or can't be. It is 50-50, 50-50, 50-50, and it is depend of a distance between genes. If distance is more than 50 mogonids, crossing over is not happen. But when the distance between two linkage genes are less than and is 39 mogonids, this process is happen. Okay, and the possibility of crossing over is depend how many mogonids are between two linkage genes. Okay. Okay. And for example, okay, and why we must know distance between genes A and B, for example, between two genes, because when we know a distance between these two genes, we know a chance of crossing over, and we know a percent of how many persons in the next generation we can see with crossing are formed after crossing over. Okay, for example, when distance between genes A and B are 3 mogonids, there is a 3% of chance 3% of crossing over, and there is 3% of all persons in the next generation. If we have 100 person in F for example 3 generation, and we have a distance, and we know distance between genes A and B in 3 mogonids, there is a 3% of chance of crossing over, and there is a 3% from 100 persons of F3 generation, and there is a 3 organisms. I hope you understand this clear. Okay, and there is all.